Hello dear students, welcome to Practical Biochemistry. The topic is estimation of serum total cholesterol by CHOD-POD method. Estimation of serum total cholesterol. Cholesterol is a steroid with alcoholic group. It is most abundant sterol in human tissue and body fluids. It is derived from exogenous diet as well as endogenous sources. Two forms that is present in the blood: free cholesterol around 30% and esterified cholesterol that is around 70%. Transported bound to lipoproteins, high density lipoproteins and low density lipoproteins. High density lipoprotein or HDL cholesterol is good cholesterol, and LDL, that means low density lipoprotein cholesterol, it's a bad cholesterol. Increase in the serum cholesterol level is one of the main factor for atherosclerotic heart disease like myocardial infarction, cerebral hemorrhage and thrombosis. Now the principle of CHOD-POD method: in the presence of water and cholesterol esterase enzyme, cholesterol esters are converted to cholesterol and free fatty acids. Now this cholesterol, in the presence of oxygen and cholesterol oxidase enzyme, it forms cholesterol 3,1 plus hydrogen peroxide. Now this hydrogen peroxide reacts with 4-amino antipyrine and phenol. In the presence of peroxidase enzyme, it forms quinonimines and 4 molecules of water. Pink color is appeared and read absorbent colorimetrically at 520 nanometer wavelength. So that is the principle of CHOD-POD method. CHOD, that means cholesterol oxidase enzyme, and POD, that means peroxidase enzyme. Reagents required for this test: cholesterol reagent, cholesterol standard solution, that is 200 mg per dl. Now the procedure: take 3 test tubes and label test tube blank, standard and test. Add 1 ml of cholesterol reagent in all 3 test tubes blank, standard and test. Add 10 microliter of standard solution in standard test tube. Add 10 microliter of sample in test tube. Add 10 microliter of distilled water in blank test tube. Mix well all 3 test tubes. Incubate at room temperature for 5 minutes. Read absorbance colorimetrically at wavelength of 520 nanometer. Now here it's the procedure. Take 3 test tubes and labeled B for blank, S for standard, T for test. Add 1 ml of cholesterol reagent in all 3 test tubes blank, standard and test. Add 10 microliter of distilled water in blank test tube. Add 10 microliter of standard in standard test tube. Add 10 microliter of sample in test tube. Mix well all 3 test tubes and incubate at room temperature for 5 minutes. After 5 minutes, read absorbance at wavelength of 520 nanometer colorimetrically. Now take your readings. Observation: OD of blank 0.00, OD of standard 0.21, OD of taste 0.41. Calculation part: OD of taste minus OD of blank divided by OD of standard minus OD of blank multiplied by concentration of standard is equal to 0.41 minus 0.00 divided by 0.21 minus 0.00 multiplied by concentration of standard, that is 200, is equal to 0.41 divided by 0.21 multiplied by 200 is equal to 390 mg per dl. So the result is concentration of serum total cholesterol is 390 mg per dl. Reference range or the normal range, total cholesterol 150 to 200 mg per dl. So we get the value of 390 mg per dl, that is high value than that of the normal limits. So this condition, it is known as hypercholesterolemia. Now the interpretation part. When the cholesterol level is above the normal value, then the condition is known as hypercholesterolemia. The causes includes diabetes mellitus, nephrotic syndrome, obstructive jaundice, hypothyroidism. Idiopathic hypercholesterolemia of unknown etiology occurs in some families. These conditions lead to atherosclerosis. When the cholesterol level decreases below the normal range, the condition is known as hypocholesterolemia. This occurs in hyperthyroidism, pernicious anemia, hemolytic anemia, malabsorption syndrome, hepatocellular damage. If you like this presentation, please try to share it with your friends, group, batch and colleagues. Thank you so much everyone.